Hi class, today we are going to learn about mitosis. And as our introduction video, we are going to listen to what mitosis is, and then we are going to answer and see which questions we would feel most comfortable answering. What is mitosis? What are mitosis and meiosis? What occurs at different phases in cell division? And what are mitosis and meiosis similar and different? How are they similar and different? So without further ado, we're going to go ahead and play the video.
To produce body cells. Mitosis. Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? One major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes, very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So, ready for the tricky part? Because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and remind you to stay curious.
Okay, so now that we've listened to what mitosis is, let's discover which question you would feel most comfortable answering. You're going to take and drag a check mark to the right one or a question mark to the questions here. In slide 3, we have our vocabulary. A cell is the smallest structural and functional unit of an organism. Our bodies are made of them. Cell division is how cells reproduce by splitting apart. A somatic cell is all cells in the body except for germ cells. Mitosis is the way that somatic cells divide, and germ cells are only found in the ovary and testes, which are the reproductive organs. And then we have meiosis, which is the way that germ cells divide. We will not be getting into detail on meiosis, as that is something you will review during your 8th and 9th grade years. Now, right here, you're going to type and tell me what is a cell. So look on your definition here and tell me what is a cell. For vocabulary, chromatin is unwound DNA found in the nucleus. Chromosomes are tightly packed DNA found only during cell division, and it can be found under a microscope. So right here you have chromatin right here, and then you have a chromosome going through the split here. What are the differences between chromatin and chromosomes? Put your answer here. Chromatid is each of the two thread-like strands into which a chromosome divides during mitosis. Sister chromatids are two identical copies of that chromatid. A centromere is a structure in a chromosome that holds the two chromatids together. What does a centromere hold together again? Put your answer right here. Vocabulary. We have spindle fibers, which is what controls the movement and separation of the chromosomes during division. Our centriole helps in the formation of spindle fibers. The nuclear envelope is a membrane that separates the nucleus from the cytoplasm in eukaryotic cells. As you can see right here, during meiosis and mitosis, we have our nuclear envelope that is starting to break down, as you can see. And then we have our other one right here where it's turning into oval shape. And then we have our centrioles right here. And then we have our spindle fibers, which is these long string spider-looking things that are developing throughout the entire process. Somatic cells are any cells that is not a germ cell. It contains 46 chromosomes in humans. It divides by the process of mitosis. And it is found throughout the body, in blood vessels, blood cells, bone marrow, brain, muscles, skin, teeth, intestines, and other internal organs. Right here, you're going to go back and look at your vocabulary, and you're going to drag and click to the right thing. So, this type of cell division that creates germ cells, that would be known as what? Mitosis or meiosis? If you said meiosis, you were correct. So you would take and you would drag it over to that box, just like that. Now, this is going to be a show what you know slide, so make sure that you work on this on your own. Cell division by mitosis. Mitosis in somatic cells results in two cells exactly the same as the parent cell. The two new cells are diploid cells. Mitosis involves one set of division stages. Mitosis creates new cells for growth and repair. After a cell goes through mitosis, how many cells are there going to be? Put your answer there. Mitosis is also used for asexual reproduction, meaning it reproduces on its own without assistance. An example is budding, vegetative reproduction, binary fission, and fragmentation. For example, some of these types can be jellyfish, worms, and plants. There's 4 basic phases in the cycle of mitosis. You have prophase, which is right here. You have metaphase, which is right here. Anaphase, which is right here, and then telophase and cytokinesis are part of the same thing. This occurs in strict sequential order called the cell cycle. It produces diploid cells, meaning two cells with the same genetic makeup as the parent cell. Interphase. It occurs between mitosis cycles. This is where it starts off with. The chromatin is unwound and the cell grows in preparation for cell division. Note the position of the centrioles. Just before mitosis starts, single chromosomes replicate and make a pair of long, stringy sister chromatids. Where is the chromatin during this interphase? Where would the chromatin be? Right here. What is this known as? Put your answer here. Prophase. It's the first phase of mitosis. Chromosomes condense and become visible through a microscope. Spindles begin to form, and the nuclear membrane breaks down. Centrioles begin moving toward the poles. The nuclear membrane breaks apart to allow the contents to be used in mitosis. What is stored in the nucleus? So, this is the nucleus right here in the nuclear envelope. What is stored in the nucleus? Write your answer here. 15. Metaphase is the second phase of mitosis. The chromosomes guided by the spindle is known as the microtubule fibers line up in the middle of the dividing cell. The centrosomes are opposite ends, which are spindle poles of the cell. The centrosome is an organelle near the nucleus of the cell that contains centrioles in animal cells, and from which the spindle fibers develop in cell division. Anaphase is the third phase of mitosis. The two sister chromatids of each chromosome are pulled apart by the spindle fibers. Chromatids move away from each other toward the poles, and now each of them are called a chromosome. The cell elongates so that the poles are further apart. So it starts off as a nice little circle and turns into oval as they start to separate. Why does the cell get longer during anaphase? Why do you think this is getting longer? Put your answer here. Telophase and cytokinesis. The last phase of mitosis. The chromosomes have reached the poles and began to unwind. Two new nuclear envelopes form around each of the two separated sets of chromosomes, forming two nuclei in one cell. The cleavage furrow begins separating the cytoplasm into two cells, each with a nucleus. This is cytokinesis, and when complete, the cell has divided into two daughter cells exactly the same like the parent cell. Here you're going to take and determine which one is prophase, which stage is prophase. Then what you will do is you will click on it, and then you will move it accordingly. You'll move your box around accordingly. See if I can. Right here, you will take and you will determine which phases these are and what order they are in. This will be a show what you know slide. And finally, our last slide is mitosis was named in 1882 by German scientists. The name was taken from ancient Greek word mitos, which means string. What parts of the mitosis may have inspired that name? So, right here, which part shows the strings? Which part would it be? You can refer back to let's see here. You can refer back to this slide, slide 6, to answer that question. And that is all for now.